So should we do this like a first we'll have the presentation, like then we have a discussion session, or how does?
Yes, we can have presentation and then discussion with the students.
Okay. Yeah, and also we can make it this is like an interactive. If someone has the questions, feel free to stop and ask the questions anytime. So you can see my slides, right?
Yes, sir.
Great. Again, good morning, everyone. I think it's a great forum to, it's very happy to see that, where you're organizing session to, promote the innovation. I think that's the need of the hour. And today I'm going to discuss about primarily a translational science and how, multiple examples we can take to show that how innovation could be used in science entrepreneurship. Right? So even to begin with, like when you say about the translational science, right? If you see the core definition of translational science is like, identifying an unmet need and developing technology through innovation to solve that unmet need, and which can be translated into the clinic to reach to the people to improve the quality of lives of people. That's a classical definition of translational science, what we use for. And but like if you see, right, this is the classical depiction of like a valley of death that exists in the clinical translational research. For example, when you talk about it's irony that like a majority of the scientific innovations and breakthroughs, they remain in the laboratory. They are not able to cross this valley of death to reach to the people, right? But the positive note is this gap can be bridged through productive collaborations between basic scientists and clinicians and entrepreneurs. More importantly, even after developing this innovative science, the breakthrough science, that need to be converted into viable products to reach to the people, right? So that's where the entrepreneurship practice plays a major role, converting your scientific solutions into viable product to reach to the people. But interestingly, if you see the entire worldwide, right, when you talk about the translational science, very small portion of scientific group work in the translational science. Even among them, like you see very small portion of them, they do this end-to-end journey, like right from taking idea to till reach to the market, right? More importantly, like we'll also see another trend, right? Like once a group does this entire translational journey, they then tend to repeat it over and over to bring multiple innovations to the market. So then the question comes why such a very small portion of fraternity actually practicing is, and is there any magic recipe to do this, right? So the answer is actually there is no magic recipe, but if you see very closely observe this to do whatever the examples of science, successful examples of science translational research, there are few common factors we see which plays major role in converting or practicing this highly translational science, right? So with our experience in 10-15 years in the field, and also by closely observing the world's best clinical translational scientists, and I could formulate a 4-step rule. These are just my observation. In my opinion, I think there are 4 common threads, 4 common points we keep seeing in any successfully translational science examples what we see. I thought I'll just share those thoughts with you. And of course, again, this is just my point of view, so you can take it with a pinch of salt. In the journey, right, very first step, what we need to have having a great idea, right? But it's it's very subjective, right? What is great idea? Because we all think that the idea is what we get the great, right? No one says hey, I have a bad idea, let's go and pursue it, right? So then the question is how an idea qualifies as a great idea. So in a translational science or in an entrepreneurship, idea is which addresses to huge unmet need. Okay, so the quality of idea not only decided based on the intelligence of idea or a complicated science, right? Rather, whether it is addressing a huge unmet need or not. Okay, that is your benchmark to qualify whether your idea is a great idea or not. Just to tell you, right? For example, say you can use very complicated, very intelligent scientific idea, biological phenomena you can discover. Through that, say you develop a drug for headache, right? So in terms of science is top notch and all that. But if you think about as a translational science, whether does it qualify as a great idea? The question is, because if today you can just walk to the pharmacy and you can get 10 different type of medicine for your headache, right? Then the question comes why one need to put the entire research to develop another medicine for headache, right? So how much extra value that adds to, that is what decided, right? But instead of that, for example, you say if you develop a drug for, say, sepsis, where absolutely no therapy is available today, in that case, even 50% of success comes through your idea is a great idea, because that adds huge improvement in the field, huge leap forward in the field. So that's why when you think about ideas in the translational science, one need to think about or ask the right question, what problem it is solving and how unmet need it is, right? So that's where it qualifies what is your idea is. And your ideas can be lead to two types of solutions, right? One is one product of technology, or you can develop a platform technology. Say, for example, when you say one product of, you have identified the problem, now you can develop the technology which can exactly solve that problem, right? That's great. It works very well. But if you're thinking about forming a venture, or if you're planning to translate that into clinic, it is always advantageous to develop a platform technology. What do I mean is, say you have a problem, you identified one problem, now you develop the technology. With that technology, you can solve that problem. But just by tweaking around your technology, and if you can address multiple other problems, then that gives you a lot more scope, right? For example, you just take if you're developing a drug, cancer therapeutics, so one can develop, say, one particular drug for one particular type of cancer, say breast cancer, for example. If that works for only that particular cancer, it's great. It's still you're solving problem. But the similar platform drug, if it can also be used for treating other types of cancers, like pancreatic cancer, colon cancer, and so on, then your scope of the translation goes very high, and it also attracts a lot more attention and funds and all to take to the next level. So that's why one need to focus developing platform technologies if it's possible to convert them, your ideas, into actual realities. Then the second step, having a broad patent, right? And it is absolutely critical when you're thinking about translational science, because your intellectual property is very key. How strong IP you have decides how your innovation is, right? So, but getting patent applications is not very easy, right? Because if you have the incremental idea, it doesn't really generate the patents, right? So it has to be completely out of the box, innovative ideas. And that's where, as innovators, we need to be super critical about our work itself, to see whether whatever the idea we develop, is it a merely extension of the idea what has been done earlier, or is it just an incremental change, improvement compared to what is existing, or is it a completely novel which has not been done and which people couldn't think of before and it came up with. So this is where one has to be super critical, so that one can develop really strong patent portfolio. And this patents also like not only just broad patent, it should be a blocking patent, right? Because as you know, patents, there are two types, right? Like two advantages of the patent. One, it will allow you to practice the invention what you have made, but at the same time, it will also prevent other to practice the same, right? So that's absolutely critical as well. And then the question is how one can develop these patents. And I really urge people to learn some basic writing, patent writing skills, because the patent writing skills is completely different than writing your papers, research publications, or reviews. So, and how you develop the skill is like you need to be like have super understanding about the prior art, in the sense like whatever ideas we are working on today, right? All these problems have been worked before, like several attempts have been made by people. We should know everything about that. What all people have tried and what worked, what didn't work, and how our current work is going to be different than that. So that entire broad spectrum idea, comprehensive idea people should gain, then only we can develop really strong patents. Okay, so, and the third one is having a publication in top-tier journals, right? Again, like this is a little bit subjective, right? As a scientist, like, I think we should judge the quality of research or the quality of science merely based on the, the quality of the just science itself rather than where it is published, right? So, but then the question is then why one need to try for like, better journal or top tier journal. So when you push for that, there are two advantages we get, okay? One is the credibility, right? So in the sense like, when you have these top tier journals and they have set it up very high rigorous peer review system, so that like, they already set up a benchmark, right? Your innovative level should be the top of that, then only it reaches to that stage. So when your work gets published in such journals, it's already validated, vetted by the rigorous peer review systems that people already know. It already crosses certain standard of the research. So people tend to believe that more. And you see, if people are working in your field, no matter where you publish, they can understand and they can appreciate your, right? But like if you're thinking about translating that, if you're thinking about like, going for, tomorrow if you plan to go for forming a venture, you have to raise the money and so on. And all of them are like, they are not like, domain expertise in your field. But like, most of them are like aware of the standards of journals and like, where it gets, and, people can start having the belief. And at least you'll get the first meeting easily when you have such publications based on your innovation. And second is visibility, right? Again, like, when you publish us in top tier journal, various media tend to, focus and tend to pick up those ideas and write about it, so that you will have much wider readers, right? And the last point is having a great team, okay? This is like, it's all about people and people and people and people, right? In fact, if I say as a priority wise, I would keep team as the first than anything else, because, you need to find a right set of people, because you can take a great idea, but if you give a bad set of team, they can kill it overnight, right? Easily. But if you have a right set of people in a team, even 10 ideas fail, they can work and they can make sure that your 11th idea will be successful, right? So that's why it's super important to identify a team which can have the same vision as you have, and the same dedication as you have to take to the forward. And also, like, if we are talking about science entrepreneurs, like, we are trained in science and we are trained in research, and we might not have all other skills, the complementary skills, like, in a business or business development, marketing, or like there's so many other skills you need if you want to become practice science entrepreneurship. I think one is learning all those skills, but like which is like super time taking and like, not everyone can grab that. So the best is find the domain expertise people in that area who are much better than you and collaborate and form a team to take to the forward. So these are the 4 common factors we keep seeing in all successful translational science examples, what we see. So then, what I thought is like, with this brief intro, then I thought like I will take some of our own research examples and I'll walk you through what is the research we have been doing and like where they are, so on. Okay, so my lab is primarily focused in clinical translational research. We are located at inStem, and where my lab is 2 major areas we focused on. One is biomaterials and second one is chemical biology program. In the biomaterials, what we do is like we develop variety of bioengineering concepts and biomaterials to improve the efficacy of the existing drugs by eliminating all the limitation what they have today. And whereas in chemical biology program, it's a new drug discovery program where we develop completely novel drugs. Okay. And as name suggests, like again, biomaterials can be like 2 types. One is therapeutic biomaterials and prophylactic biomaterial, which means like, the biomaterial which can be used in the treatment of the diseases, or you can also use them to prevent the diseases to occur. Okay. In the drug discovery program, there are again like 2 types we are doing. One is new chemical entity, where the classical medicinal chemistry based drug discovery we do, and also novel biologics based drugs also we develop, like such as like proteins, enzymes, and cytokines and so on, used as the drugs. Okay, so to begin with, like we can discuss about our efforts in the therapeutic biomaterials, which could be used for multiple medical applications. As you know, like, inflammation is inherent part of most of the diseases, right? Like, such as arthritis, psoriasis, lupus, and so on. But today, if you see, like, despite having many potent anti-inflammatory agents and immunosuppressive agents, the overall therapeutic outcome for the treatment of these diseases are really poor. Okay, so the main reason for it is like, if you see the level of disease, that means the level of inflammation in these diseases is highly fluctuated. For example, if you see here, it's never like a disease is never like a steady state, right? You have like increased period of disease, and you have steady state, and you have occasional remissions and occasional flare-ups and so on. And unfortunately, we do not have a non-invasive way to measure the level of disease a patient has today. Okay, so because of that, it becomes super difficult for the doctor to prescribe the drugs, whether what is the doses one need to give and how frequently these doses should be given, right? So that's where we thought, if we can develop a novel drug delivery system to solve this problem, then we might have a huge impact, because if you can target inflammation, right, as I mentioned earlier, then it becomes like a platform technology, because inflammation is the inherent part of so many diseases, right? So then if you can tackle inflammation in a right manner with a supreme control, then this could be used for multiple diseases, for the treatment of multiple diseases. So then the question is like, what is the ideal way of delivering drugs for this inflammation, guys? Typically, if you see like almost 70 years history of the drug delivery, there are 2 major concepts have been developed so far. There's like 2 breakthrough concepts, right? Very early in the field, identifying the excipients. These are either small molecule based or the polymer based excipients, where using these appropriate excipients and drug combinations, one can solubilize the drugs and give the drugs. With a single dose, if you give them, it keeps the drug in a therapeutic window for a certain period of time. When it comes down to the subtherapeutic level, you give second dose of the drug, and that keeps for some other time and so on, right? That's how, like, your clinician suggests you whether you have to take a medication once in a day or twice in a day or so on. And this is great for if a patient has to take drugs for like, few days to a week or so. But like when people have to take this medication for the chronic disorders, where people have to take like several weeks to several months, we tend to miss the doses, right? Either we miss because we cannot take or delayed manner, or we cannot take more frequent than what is supposed to be taken, because it becomes like a super toxic or inefficient. So that's why, like, to overcome these limitations in late 70s, people have come up with the sustained drug delivery concepts, where people have developed variety of biomaterials where you can encapsulate the drugs into these biomaterials, and with a single dose administration itself, they start sustained manner, they start releasing the drug and keep the drug in a therapeutic window for a long period of time with a single administration itself. So in that way, like, one can treat the chronic diseases efficiently. So delivering both ways of these drugs actually revolutionize the way you treat the patients, right? But as I mentioned, unfortunately, if you see the inflammatory diseases, the level of disease itself so fluctuated, it becomes super difficult. Either way of delivering drugs won't be efficient, because either you end up overdosing the patients or underdosing the patient, right? So then what is the ideal way of delivering drugs, right? That should be like, your drug delivery should be the mirror, identical to the level of disease a patient has. That means whenever the disease comes, it should release the drug. If remission period, stop releasing. If flare-up comes, it should release, and so on. But as I mentioned, we don't even have the technology to measure the level of disease a patient has. So then it's impossible to impart that knowledge and fine-tune the material externally to release this profile, right? So the only way one can achieve this is if disease itself can control, okay, decide when to release the drug and how much dose it should be released. Then only we can achieve this kind of disease responsive drug delivery. So we thought, like, that's the concept we would like to develop. So that's where we develop this disease responsive drug delivery systems. If you see here, what happens, like, first we need to identify a disease associated biomarker. This could be like, a cytokine, an enzyme, or any biologics which is produced during the disease time. And now you develop a system, drug delivery system, which can be implantable. But in the absence of this biomarker, it doesn't release the drug. But when the biomarker comes in contact, that should able to stimulate and release the drug in a responsive manner. And the released drug goes and alleviates the disease, and that stops the production of biomarker, and that stop releasing the drug. So in this way, the disease itself is controlling when to release the drug and how much to release the drug. Okay, that's the overall concept what we have been developing in our laboratory for the past 6-7 years. And I will show you like how this concept could be used for multiple medical applications, such as like protecting transplanted organs, and how we can use for the treatment of arthritis and IBDs and so on. So when you talk about organ transplantation, there are two types, right? One is like a solid or internal organ transplantation, and where you see like life-saving organ transplants like lung, liver, kidney, and so on. The second type is called vascularized composite tissue allotransplantation, where instead of like a single solid organs, like your entire composite tissue, like for example, your full hand, full leg, full face can be transplanted from one person to another. And here, if you see, each color-coded part has already been successfully transplanted, which means like potentially every single part of our body can be transplanted from one person to other, right? So then today, like I'm going to talk about specifically about the hand transplantation example, because if you see, like, the microsurgery has developed so much that now people can do successfully the single hand transplants and double hand transplantations very easily, right? And the post transplantation, and these people can actually regain their function and they can start doing the normal chores nicely. So this is incredible, right? Then the question is everything is okay with these patients, right? But unfortunately, answer is no, because as you know, like our immune system is like our friend, right? It is designed to identify, a foreign material and foreign pathogens and neutralize them. That's our primary immune defense system. But in these transplanted patients, like your friend becomes foe because your tissue that is like a organ that is transplanted from other person, it is recognized as a foreign material and your body tend to reject it, right? So that's where to control your immune system, these patients has to take like, large amount of immunosuppressant drugs and throughout their life to protect themselves. But unfortunately, that leads to huge side effects because of this long-term use and systemic use of immunosuppressant leads to problem called global immunosuppression. That means their entire immune system get compromised. Even a small infection could be very lethal for these patients. Potentially a flu might able to kill them. So that's where like, one need to develop a system like where you can protect the transplanted organs without causing your global immunosuppression and so on. Again, when you take systemic manner, large dose of the drug every day, that has like a severe other non-specific toxicity. For example, like almost 90% of the iron transplantation patients comes back with a kidney failure within 3 years of the surgery because of the immunosuppressant what they take. They're highly renal toxic. So that's why like one has to develop the systems where instead of much more systemic manner, instead of that, like one has to develop this local therapies and intelligent therapies to one is to prevent all the other non-specific organ toxicity and prevent the global immunosuppression to protect the graft. Okay, so then we thought like, having, because if you see again, like when you have this immune activation that leads to the inflammation, again, inherent part of this disease, right? So we thought having this inflammation responsive material could lead to developing this better therapeutic strategies. So, and it is known that like, during inflammatory like condition, they produce high concentration of these proteolytic enzymes, matrix metalloproteases and esterases and so on. So what we thought is like, if we can develop a material which is stable in the body in the absence of these enzymes, but like when these enzymes produced by the inflammation, they can degrade, then we might have this disease responsive characteristics of this material. So then our approach is like, we design like a small molecule. These are amphiphilic molecules. Like, we have hydrophilic, hydrophobic part of the molecules, which can self-assemble together to form this nanostructure. And during this self-assembly process, one can encapsulate the choice of drugs. Here is immunosuppressants. And this self-assembly should be robust enough that they shouldn't release the drug in a non-specific manner. But when they get exposed to the enzymes that are associated with the inflammatory like condition, they should be able to degrade these amphiphils to release the encapsulated drug in a responsive manner. So that's the overall approach what we are taking. And then the question comes, what kind of small amphiphils one need to make? As an organic chemist, we can make like a beautiful new molecule. But what we thought is, instead of that, we focused on a group of molecules called GRAS agents, generally recognized as safe agents, because these are the compounds have been used in human products extensively, and their safety profiles are well known. And what we have done is we screened several hundreds of these molecules and narrowed down to 4 molecules which has the ability to form these self-assembled structures. One of them is triglycerol monostearate. It's a simple amphiphilic structure, but it has a fine balance of hydrophilic, hydrophobic moieties to promote the self-assembly, and also having this inherent ester group can be degraded by the proteolytic enzymes that are associated with inflammation. Then what we have done is we took this tacrolimus. Tacrolimus is our known immunosuppressant drug, which is prescribed for the hand transplantation patients. And we optimize conditions to encapsulate within these gels. And we develop this injectable hydrogels where encapsulated with this immunosuppressants. Then once we have these gels, like we went and tested their ability to release the drug in response to the proteolytic enzymes. As you can see here, in the absence of any proteolytic enzymes, you don't see any release of the drug. Like we have non-specific 5 to 10% of the drug release, but it stabilizes. We tested even after 90 days, like 3 months, we don't see any non-specific release. But in the presence of enzymes, you can see nicely they degrade and release the drug. These are like a nicely stabilized in vitro enzymes, right? Then we wanted to see whether do we get like a similar response for the in response to inflammation as well. So to test that, we took the macrophages, which are like our immune cells, and you activate them. And when you activate them by LPS treatment, and this activated macrophages resembles your inflammation-like condition in the body, and they're known to produce these proteolytic enzymes. Then we wanted to see these in situ generated proteolytic enzymes can degrade the gels to release the drug. To test that, we took the conditioned media from activated macrophages and as well as a non-activated macrophages as a control to see the release of the drug. When gels were exposed to these conditioned media, as you can see, when exposed to the non-activated, meaning like a non-inflammatory like conditioned media, you don't see much release of the drug. This is as good as like, buffer, right? But whereas when you expose to the conditioned media that was taken from the activated macrophages, you can see the release of the drug. Not only that, whenever you give fresh pulse of activated conditioned media, you see the responsive manner they release the drug. So which is clearly suggesting that indeed this material has the ability to respond to the inflammation and release the drug in a dose responsive manner. So once we have that, then we went and tested their ability to protect the transplanted organs from the rejection, right? So what we have done is we took completely two mismatched rats from like completely from a brown Norway rat to Lewis rat. We did the entire hand limb transplantation, and the post-transplantation surgery, we can take these gels which are loaded with the drugs and inject them as a subcutaneous depose under the skin and follow the survival of the graft. Okay, just to quickly summarize the main results, like you see, once you do the surgery, if you don't give any treatment, or you can inject only gels without having the drugs, okay, within 11 days, 50% of the animals reject the graft. Okay, as you can see, severe inflammation in the tissue and grade 1, grade 2 rejection in these animals. Now, as a control, what we do is like a take only drug, tacrolimus, and without encapsulating into do the local injection. Okay, in that case, you can see like within again 33 days, they start rejecting the graft completely. Okay, and we can see severe muscle tissue damage and like high inflammation and grade 3 rejection. We can see here, right? Now, the same amount of drug you take and encapsulate in the gel and give one single injection the post surgery. Okay, in that case, if you see, you can see like no sign of inflammation at all, and hair grows back normally, and like even after 100 days, we don't see not even single animal reject the graft at all. And not only that, like this animal start gaining function, and now it is able to move its limbs and so on. And then we have tested their entire systemic inflammation, immune system, it is active. And we have tested their all other organ toxicities. Absolutely, there is no non-specific organ toxicity and so on, which is clearly suggesting the advantage of having this disease responsive or inflammation responsive drug delivery in protecting the graft. Then we wanted to see how long this can actually protect the grafts, right? So as I mentioned, like if you post surgery, if you do one single injection, just only drug, the survival is up to 33 days. The same dose, if you put it in gel and give single injection, survival went to 190 days. Okay, and then now then we have done extensive pharmacokinetics to see the drug availability and so on. Based on that, if you do periodic injections, like meaning like after surgery, day 1, you give 1 injection. After 3 months, you come back and give 1 more injection. Survival goes to 260 days, and without having any sign of other side effects at all. And gold standard in the field today is if you can cross 60 days survival without rejection, that is the gold standard today in the field. And here we can see even, and 60 days even with the other side effects, right? But here, like, we have a system which can, periodic injection can give the lifelong protection without having side effects. So that's a huge improvement what we can see. And after that, like, we wanted to see whether, as you know, like, when people take immunosuppressant, they are prone to have opportunistic infections, right? Because their immune system is compromised. Then we wanted to see whether having this technology, whether we can prevent such opportunistic infections. So to test that, what we have done is, like, again, 2 groups of animals were transplanted. One group of animals received, these periodic injections, only 4 injections every 70 days once. And results were analyzed in 280 days. We terminated the study. But whereas in other system, these animals, much smaller doses of drug was given, but every single day, like a systemic therapy, like how patients take today, right? In both cases, if you see, the survival of the graft is same, okay? But more importantly, if you see, the animal that took received the systemic therapy, like what patients take today, all of them developed opportunistic infections. And whereas none of the animals that received this disease-responsive biomaterial therapy, none of them developed an infection, which is clearly suggesting having the advantage of this enzyme-responsive or the disease-responsive biomaterial-based drug delivery systems in protecting transplanted organs. So of that, we went and evaluated their efficacy in larger animals as well. In pigs also, it is working beautifully well. So now we are putting clinical studies together. So most probably we'll be going to the clinical trials in the next few years or so. So with that, I can come to the second examples where, like, how we can use this localized drug delivery systems that are inflammation responsive biomaterials for the treatment of arthritis. As you know, arthritis primarily affects all the major joints, right? And but today, if you take all the powerful anti-inflammatory drugs like COX inhibitors and so on, these are taken in systemic manner, and but one has to take large doses of these ones in order to have the effect at the joints, because large doses needed. But because of this large doses, it has like a plethora of side effects. Okay, it causes severe systemic toxicity. Because of that, multiple drugs like Vioxx and Dextra was recalled from the market by FDA because of their systemic toxicity. So to avoid this, then people have started injecting the drugs directly into the joints through intra-articular injections, right? Although it is little effective, but unfortunately the overall therapeutic outcome is very poor, because these are free drugs, right? When you inject them, they remain for some time and they diffuse out from there, right? And the maximum retention time, if you see here, very less. And there are series of drugs have been tried to do these local injections, but their average time to be 0.1 hours to 5 hours maximum, they can remain there. And according to clinical protocols, we can do only 2 injections per year. Otherwise, we end up damaging the joints because of the injections, right? So that's why then what next people thought is, instead of having the solubilized drugs, they started encapsulating these drugs into variety of biomaterials, you take nanoparticle, microparticles, hydrogels, and so on, vesicles, and so on, and they started injecting localized manner. But again, unfortunately, there wasn't better therapeutic efficacy. The reason is, as I mentioned, the inflammation is highly fluctuated, right? And it comes and goes. And these delivery system, despite whether, irrespective of whether the patient has inflammation or, they release the drug for a certain period of time and they go. So because of that, you're delivering the drugs when it's not needed. When disease comes back, you don't have the drug to trouble with. So that's where we thought having this enzyme responsive manner might have the great advantage to treat this arthritis. So again, we took this TA, triamcinolone acetonide, which is prescribed drug for arthritis, and we optimized conditions to encapsulate into these hydrogels. And again, they have shown similar manner. This drug also released in response to the enzymes and also in response to the inflammation, as I mentioned earlier. And but more importantly, we wanted to see, because when you see in arthritis patients, your joints are completely inflamed, right? Their joints have the synovial fluid. Your entire synovial fluid becomes inflammation. So we wanted to see whether such inflamed synovial fluid also can release the drug in a response to the inflammation. So to test that, what we have done is we took this conditioned media from arthritis patients as well as healthy volunteers whose synovial fluid doesn't have the inflammation. Okay, and those synovial fluids were incubated with our gels and tested the release of the drug. As you can see, if you see this yellow line, where when the gels were exposed to the synovial fluid that was taken from the healthy volunteers, the release wasn't great, because the release is similar to what we observed in the PBS, right? That means your synovial fluid is not really promoting any additional release. But whereas when the synovial fluid that was taken from the arthritis patients, when exposed to gels, you can see the release of the drug. Okay, and again, like whenever you give fresh pulses, like it keeps increasing the release of the drug, and which is clearly suggesting that the inflammation that is present in the synovial fluid in arthritis patients can trigger the release of the drug in an on-demand manner. But whereas if there is no inflammation, this might not release the drug, right? So then after that, we wanted to see, for example, if you don't have an inflammation, right, whether or the gel that is injected into the joints, can it remain there without releasing the drug? And whenever inflammation comes, will it be able to release the drug? So to test that, what we have done is you take healthy animals which don't have any disease, and we fluorescently label these gels and inject it into the joint. And over a period of time, like a couple of weeks, if you see, you don't see much reducing in the signal, and this signal is localized in the joints, which is clearly suggesting that if there is no inflammation, the gels are still there and they're not releasing the drug, right? And despite of this mechanical strength, right? Because these animals are keep running around and walking around, so you have the mechanical stress. Despite of that, you don't see the depletion of signal. Now in these animals, we can induce the arthritis, a mild arthritis, by injecting 37.5 microliters of serum. Because when you inject this serum, this concentration, it is known to form mild arthritis from day 4 to day 5 onwards. Okay, as you can see here, after 4 or 6 days, you can see the rapid dissolution of these gels and release the drug, right? And but if you increase the dose of this, double this dose, and that increases and it produces the severe arthritis from day 2 onwards. In that case, if you see the animals that received high doses of serum from day 2 onwards, it's rapidly dissolve the or degrade the gels to release the drug. So which is clearly suggesting that these gels have the ability to remain stable if there is no disease, and withstand all the mechanical stress. But whenever the disease comes, and it can able to respond and release the drug whenever it is required in an on-demand manner. So once we proved that, we went and tested their overall therapeutic outcome. So again, we induced the disease, and then there are 2 types of injections were given. One, either free drug was given without encapsulating into the gels, and the same dose of drug was encapsulated in the gel and given. In that case, like if you measure the overall therapeutic outcome, one way to measure is the swellingness of the paw, right? So the less swelling is better treatment. As you can see, the drug in the gel has much better therapeutic outcome compared to the same dose of drug given freely. And the same trend you see when you have the clinical score as well, right? So in an overall, it is clearly suggesting that having this inflammation responsive material in a localized manner can improve the treatment of IBDs, right? Arthritis. That brings to the third example where we can see that inflammation targeting microfibers for the treatment of IBDs. As you know, IBDs comes in two variants. Like one is ulcerative colitis and Crohn's disease, where small and large intestine gets inflamed. But the question is like it's not like entire colon or the small intestine becomes inflamed. You have patches of inflammation throughout your intestine. So, but like today, there is no way to release the drug or deliver the drug to the only inflamed site, right? Even when you have healthy tissue, also get the same dose of drug when you take orally, right? And because of that, and unfortunately, the side effects of these drugs is like if it goes to the healthy tissue, that causes inflammation, right? Basically, then you're turning bad tissue into good tissue into bad tissue. That's why the overall therapeutic outcome is poor. That's why these people have to take several years. Or 6 months to years, people have to take medication for many of these diseases. So ideal way of treating this IBDs would be if you have a material which can be stable in your GI tract, but doesn't interact with your healthy tissue, but it goes and finds to only inflammate tissue and specifically adhere there at the site of inflammation like a glue and release the drug to the only inflamed site. In that way, you can treat only inflammation tissue, but your healthy tissue never see the drug, right? Then you will have much better targeted therapy, right? But the question is how one can induce such specific adherence and targeting capacity into this material. So interestingly, if you see, when you have say this is inflamed tissue, our body as our defense system produces this high concentration of antibacterial peptides and bactericidal proteins to prevent the infections at the site of inflammation, right? And all these materials, if you see, they are cationic in nature, right? These are cationic peptides, cationic proteins. So because of that, your entire inflamed tissue becomes like positive charged bed, right? Then we thought can we use a simple ionic interactions, Coulombic interactions, that means you have a particle which is a complementary size charge can able to because of the anionic cationic interactions can go and bind specifically to the site of inflammation and release the drug. Okay, that's the overall concept what we want to test. And again, we selected another amphiphile. And this amphiphile again should able to self-assemble to form these microfibers and should able to encapsulate drugs, but it's also should able to render the charge to the overall assembly structures. Okay, and they should able to release the drug in response to inflammation. So in order to fit all this design criteria, we select ascorbyl palmitate. Because this is nice amphiphile which can promote self-assembly, but it can also give the negative charge and it can be enzyme responsive and so on. So once we then we fabricated this material and you can see the micron size fibers and we loaded with the gels and dyes and so on. But interestingly, if we see all these fibers, they have this high negative surface charge, -40 with the zero potential measurements you can see. So again, they release the drug in response to the inflammation and so on. Then we wanted to see whether these material has the ability to bind to the proteins that are expressed at the site of inflammation specifically, right? So then what we did is we took the transferrin is known to produce at the site of inflammation. We took this transferrin and coated with the glass plate. And as a control, we took the mucin, which are all your healthy intestine, the mucus has this mucin, right? So we coated that with the plate. And then these fibers, which were encapsulated with the dyes, were incubated and washed extensively and measured the amount of fibers they have attached. As you can see, these fibers are completely attached to the transferrin protein, but whereas they don't bind to the mucin protein at all, which is not very surprising, right? Because you have anionic particles and you have cationic protein, they tend to bind, right? So then the question is whether can we get that level of specific adherence in the living system in vivo, right? So to test that, what we have done is you take the normal animals and give them a chemical called dextran sodium sulfate for 7 days, and they develop colitis. Okay, they develop IBDs, and the colon gets the patches of inflammation. Now we provide the material, this dye-labeled material you give them. Then after that, like after 24 hours giving this material, you can surgically remove the colons and image them to see how much material it is being adhered. Okay, if you see here, these are the colons that were collected from healthy animal. Okay, we don't see any sign of fluorescence particle here, which means all the particles that were administered are washed away, because like you have high bowel movement, right? And there is no inflammation, there is no adherence. So that's why we don't see any particles here. But whereas all the colons that were taken from the diseased animals, you can see high concentration of particles in a patches manner, they adhere there. And this is even after 24 hours, despite of your high bowel movement, you clearly see the particles adhering there, which is clearly suggesting that this material has the ability to go and specifically bind to the inflamed tissue so that it can have the localized manner release of the drug. So once we have that, then again, we went and tested their overall therapeutic outcome. Again, like we took this same dose of drug was given into this particle, targeting particle versus free drug. We can see the overall therapeutic outcome is much better when you give this in targeted manner. But more importantly, we also wanted to see, although we have shown that this can adhere to the mice tissue, whether we wanted to see whether does it have the same level of specific adherence to human inflamed tissue as well. Okay, to test that, what we have done is the patients who were suffering from IBD, from the patients we took the biopsies through colonoscopy. We took the tissue from two sides. One is where they have the inflammation tissue, but from the same patient, we also collected from the healthy tissue, healthy mucus also we have taken. And outside ex vivo, we have incubated with these fibers and extensively washed and see which material has the better binding capacity. Okay, as you can see here, this is the normal tissue. You don't see much amount of fibers attached, but whereas when this inflammation site tissue, when you collect it, you can see high amount of fibers are attached there. And not only like overall, but if you see single patient wise, for example, if you take this patient in a normal tissue, say if you have X amount of adhesion, and the same patient, like when you collected with ulcerated inflamed site tissue, they have much higher adherence. And the same manner, every single patient has much higher adherence in a inflamed tissue compared to the normal tissue. So which is clearly suggesting that this material has the ability to go and identify and adhere to specifically to inflamed tissue to have better therapeutic outcome. Okay, so just to summarize this part, like having this disease responsive biomaterial as a platform technology, we could show that this could be used for the treatment of or protecting transplanted organs from rejection episodes and increase the lifetime of transplanted organs. We can have much better therapeutic outcome in the treatment of arthritis and IBDs and so on. So, but then if you just come back and if you check our our four-step criteria, does it really meet, right? So the question is it a great idea? Yes, I think each one of them have the huge unmet need. And whereas you really needed the technology to increase the therapeutic outcomes, and each one of them produce several patents applications, and all of them also produce really good publications and so on. And also we could able to find the people, right set of people to work with us to form the companies and so on. So based on that, like we put all this technology together, and 5 years back, we formed a startup company called Alivio Therapeutics in Boston. And now till to date, like this is one of the fast growing company in Boston, in a biotech company. In fact, we just made a huge partnership deal with one of the big pharma for almost like a $300 million deal they have made, where we are working with FDA to go for the clinical trials very soon with that. Okay, so just I want to know, like, how are we doing with the time? Like, do we have more time to?
Yes, yes, sir. We can continue, sir.
Sorry?
We can continue, sir.
Okay, sure. Thanks. So, so far, like whatever I mentioned is these are the therapeutic biomaterial, right? Like could be used for the treatment of the diseases. Now I'll give you one or two examples where we can use this prophylactic biomaterial, in the sense like where we can use this biomaterial to prevent the disease to occur, right? So one of the example is like, developing this prophylactic biomaterial to protect farmers from pesticide-induced toxicity and mortality. As you know, like, pesticides are neurotoxin. They are designed to kill the pest, right? Typically what happens is like, we have this acetylcholinesterase enzyme, which plays major role in our neuromuscular functions. And by controlling the concentration of neurotransmitter acetylcholine. But when the pesticides are like, there are different classes of pesticide, right? Organophosphate, organochlorines, and carbamates. They are designed to go and bind to this acetylcholinesterase in an irreversible manner. And so it inhibits its entire function. And when you have this quantitative inhibition of this enzyme, that leads to the paralysis and subsequently death. That's how a pest dies when they're exposed to pesticides. But unfortunately, these chemicals do not discriminate between pest and human, because human also have the same enzyme, and same acetylcholinesterase plays major role in our neuromuscular function as well. That's why when we get exposed to the pesticide, we also have same level of toxicity and severity, right? So just to, if you see this is the common scenario what we see, right? All the farmers in India and developing countries, they use these hand pump machines and they spray pesticides and almost without any protection, and it falls there on skin and they inhale. It's almost like walking under the poisonous rain, right? And they get like a severe exposure, and through inhalation and dermal routes, they get exposure. And when they get exposure, that leads to the severe toxicity. So, but like, again, like if you see all the literature, what has been given in the field, people say that, like, people have to get exposed to several years, and like on a chronic manner, people might get some side effects and so on. But like that was like we couldn't comprehend that because organophosphates being such a highly neurotoxins, not having acute toxicity is like it's impossible to assume that, right? So that's where what we thought of launching a survey and to understand what is the ground reality. So what we have done is like we launched a survey, like we visited almost 60 villages in Telangana state, and for each one of them we spent like almost 4 days with their field to understand what kind of pesticide they use and what kind of farming practices they have now, and what is the level of exposure they have. Do they have any? Do they feel any side effects and so on? I think that had changed complete the perspective of what is pesticide toxicity in this, because majority of the people, like almost all of them, like they have acute toxicity. The same day they get exposed, they suffer with like vomitings, a fever, muscle pain, diarrhea, and when it goes to the high dose, it can lead to the convulsions and so on, right? Just to give an example, like, a couple of years back, you might have heard about, like, even in one district, within the span of like a few months, more than 60 people have died just by spraying this pesticide in a cotton fields, right? And this is just one-time example, and this keeps happening year and over and over and over, right? So that shows the level of toxicity people can get exposed to and also it shows the lack of any technology to protect these farmers. So that's when we thought we should develop the technologies which can completely protect these farmers. So as I mentioned, this pesticide, actually they are designed to penetrate through the skin very efficiently. So they can, when they fall on the skin, they go through the skin and they go and inhibit this enzyme. So what our idea is because earlier people are trying to develop some barrier creams, physical barrier creams, so that it cannot penetrate, but which wasn't successful at all, because these compounds are designed to be super penetratable compounds to go through, because the pest, for the pest also it has to go through the skin, then only they kill, right? That's why they are super penetratable. So that's where what we thought is, instead of simple barrier cream, we wanted to develop a chemically active gel. And this is like a chemical scissor, okay, which can covalently attack the pesticides and deactivate them and break down into non-harmful products, so that it can no longer can inhibit the enzyme. Okay, that's the overall idea. So then the question is what kind of amplifier one need to make, right? So because if you see all the organophosphate structures, right, these are basically activated esters, right? And because all of them have the really good living group, so because of that you have high activity. So then what we thought, a simple, we can use the fundamental nucleophile associated hydrolysis reactions. If you break down the organophosphate ester to a phosphate, then this can no longer have the toxicity at all, right? So then that's where we wanted to develop a really good nucleophile. And then we chose supernucleophile or alpha nucleophile as a catalyst, because as you know, alpha nucleophile is the one, when you have the alpha atom, when you have the lone pair electron, the nucleophilicity goes very high, right? So based on that, there are several families of the catalysts are available, right? Like oximes and superoxides and so on. So among them, we focused on the oximes, and just to put a simplistic manner, we designed this pyridine aldoxime based nucleophile. But if you just take pyridine aldoxime, it can penetrate through the skin. So that's why we connected to the polymer chain. Okay, so this can attack with any pesticide that comes in contact and break down, and this also can regenerate itself, so that it can hydrolyze in a catalytic manner, right? So this is the synthetic scheme. You can make this quaternized pyridine aldoxime, which can be covalently connected to a chitosan. And now this chitosan, which is attached with the pyridine aldoxime, can use other excipients to form the gel. These are the polyoxime gel, okay? And as a control, we wanted to see what is the role of this nucleophile. So what we have made is just took the chitosan, again, I use these excipients to form the control gel. This is just physical barrier gel, whereas the polyoxime is the chemically active gel, right? So once we have that, we went and tested their ability to break down these pesticides. Okay, so what we have done is, this is called Franz diffusion experiment. So where the bottom chamber filled with rat blood, the rat blood contains this acetylcholinesterase enzyme, and then from top you added the pesticides, and we have a small membrane here, and then here we kept either directly they are exposed to the pesticide or through this control gel or active gel. Okay, and we measure the activity of the acetylcholinesterase. As you can see, if you take the native blood, right, their activity is 100%. But when we expose to the pesticide directly, you can see significant drop of enzyme activity. Similarly, when you have the control gel without having nucleophile, you can clearly see again it doesn't have any effect as a barrier, right? So again, you see a completely drop of enzyme activity. But when you have the polyoxime gel, you don't see drop of enzyme activity at all. That means this oxime gel is sitting there and breaking down the pesticide before it passing through, so that it can no longer inhibit this enzyme, right? So once we have this ex vivo, then we went and tested their ability to prevent in vivo exposure as well. Okay, to test that, 3 groups of animals were taken and exposed to the pesticide through dermal route. They sprayed either directly or through control gel or polyoxime gel, and the blood was collected and measured the enzyme activity there. As you can see, when you before and after exposure, the pesticide that was given either directly or through control gel, you can see the loss of enzyme activity significantly dropped. But whereas the pesticide given through active gel, you don't see such drop of enzyme activity at all. And not only in the blood, we also collected the organs, like such as brain, and we measure the activity there. Again, we can see through control gel or the direct exposure completely led to the loss of enzyme activity, but whereas in presence of the polyoxime gel is completely prevented, and which is true for every single organ we tested, which is clearly suggesting that indeed this polyoxime gel can break down the pesticide before entering into the body and can prevent the pesticide exposure. And it doesn't act as a physical barrier, but actually it's a chemically reactive gel, right? So then we wanted to see, because people get like repeated exposures in the field, right? And repeated exposure can lead to the death. Then we wanted to see whether we can prevent such lethality by using these gels. So for that, again, like 3 set of animals were given, every day 100 mg per kg dose of pesticide, either directly or through control or active gel. And again, like, here what we have done is every day before applying the, before exposing the pesticide, a fresh batch of gel was applied and given. And if you see the survival of the animals that received either directly or through control gel, within 6 days, 100% animals died. Not even single animal survived. But whereas the animal that received through active gel, 100% survival we see, not even single animal dies. And also we went and measured their blood activity, like enzyme activity in blood and tissue. Again, we can clearly see this polyoxime gel completely protects them, right? So which shows the robustness of the gel to prevent multiple exposures of this lethality. But more importantly, then we wanted to see one more surface, because whether the question comes like when you go to the field, the farmers, they apply this, they do farming very different time, right? Somebody spray pesticide for 1 hour, somebody spray for multiple hours, somebody can spray for much longer. So then the question is how frequently they should apply, right? So, or like if you have a single time application, is it enough to protect them, right? So to test that, like what we have done is, again, the 2 groups of animals were taken and both groups were given same doses for 4 days. But here what we have done is, instead of every day applying the fresh gel, only the day 0 applied only one single time this either control gel or active gel was applied. On the same gel, we kept on repeated doses of pesticide was given. Okay. In that case, again, like the control gel animal that received through control gel completely died within 5 days. Again, 6 days, all of them died. But whereas the animal that received the pesticide through active gel completely survived. So which clearly suggesting that indeed even a single application of the gel is enough. As long as gel is there, this can keep destroying the pesticide that comes in contact, because this is catalytic in nature. So that shows the robustness of the gel. So, and more importantly, like even a small amount of exposure can lead to the internal neuronal toxicity, right? Because as you know that this enzyme plays major role in neuromuscular function. This can really damage our neuronal system. So we want to quantify that. So one way to quantify is like, measuring toxicity in the sciatic nerve. Okay, how we do this? Because sciatic nerve controls your limb movements. One way to do is like you take an animal and apply like a different dyes, colored dyes on their palms and make them walk on this maze. And after they walk, you can take these sheets and measure the distance and angles and the toes they take and steps they take and so on. And all these numbers can put in one complicated equation, but we can measure something called Sciatic Functional Index, okay, SFI. What it tells is like if any animal doesn't have any neuronal damage, in a healthy animal, these numbers always come between +11 to -11, okay? And if anything goes below -20, okay, the values goes below -20 implicates the severe neuronal damage, okay? So then what we have done is like we took the animals before exposure, if measure them, and all of them, they falls into your healthy category. So there is no neuronal damage. Now you expose them directly without having the gel, and you can see there are, they can go to like as low as -60, 70. So which clearly indicates that all of them are completely exposed and they got severe neuronal damage. Now the same dose of the pesticide when you give through our gel, and if you measure them, all of them, they falls into the healthy category. That means like, the gel able to degrade completely and completely prevent the neuronal damage internally. So then after that, like, we wanted to see what is their involuntary function, because you see, one of the primary reason why these people die is, when the enzyme gets inhibited, you start accumulating your neurotransmitter acetylcholine. That means it starts stimulating your muscle function involuntarily, right? And that's why these people get shivering, high shivering, and so on. And that leads to the construction of your lungs and so on. That's how they go out of breath and die, right? So we wanted to see whether we can prevent such toxicity or involuntary movement. One way to measure is like we can measure the neuronal signaling by using this electromyogram. This is a healthy volunteer. In fact, this is a graduate student who is measuring himself, and we never got exposed to pesticide. If you see, he's not moving any hand, so on, right? So there is no voluntary signal. That's why like you don't see any real neuronal activity here, right? Now, if you take and measure same thing with a farmer he just did the farming, okay, we got exposed. Now we measure to electromyogram signal on this person, and you can see rapid signaling neuronal activity. Okay, again, like this person is not moving hand, right? There is no voluntary movement. This is just the involuntary movement because of the accumulation of acetylcholine at that neuronal junction. And that's how, like, when people get exposed, that's how you see this involuntary movements and tremors when they get exposed to the pesticides. Okay, so then we wanted to see, can we prevent such involuntary movements or signaling? So what we have done is like we measured the involuntary neuronal signaling either directly or directly exposed or through our gel, and we put first animals under anesthesia, and you measure the neuronal signaling in the hind limb, bicep femoris, and you don't see any signaling because this animal is almost sleeping. And now when you expose them directly, right, and you can see these neurons start firing, okay? And again, this animal is not moving, right? Still, you can see the firing of neurons. And whereas the same dose, when you expose through our gel, you can see absolutely no signaling. So it's as good as non-exposed animal, right? And not only under anesthesia, you can give same thing with animal that is awake. Now what happens when you get exposed to them, it leads to a lot of stress as well. So because of that, you have the combination of neuronal, voluntary and involuntary movement, because it starts shivering. So because of that, you can see these neurons are firing them, right? Now, same dose of pesticide, again, you give through our active gel, again, you don't see any non-voluntary signals at all. So which is clearly suggesting that indeed this the gel able to degrade all the pesticide so that it can completely prevent pesticide entry into the body, which subsequently can prevent the enzyme inhibition and subsequent toxicity. So after showing all that, then we developed this formulations for humans. Now a pilot study in humans are being planned now, and our whole idea is to develop this dermal cream which can be applied, which can completely prevent pesticide exposure. Because our mission is to protect every single farmers in India and developing countries from this pesticide-induced lethality. And again, if you come back and check our parameters for our translational science, right? Again, this is truly one in first in class technology, which absolutely there's no single technology exists to protect these farmers. And I think if it comes to the market, I think it will have a huge impact. And also, of course, it produced again multiple patents for us, and also good publications. And also, we put a team together to form a new company. Then we formed a startup company called Sepio Sepio Health in Bangalore, and it's been 2 years now. In fact, we're supposed to do the clinical trial this year, but due to pandemic, everything is postponed. Hopefully, we might initiate these studies in 2022. So I think that's where I can just take quickly 3 more minutes to give you another examples of prophylactic biomaterials, our recent one. That is our germicidal antiviral mask to protect from viral infections. Because as you know, once pandemic has started, and we started thinking about what can we contribute to tackle the current situation. And as you know, once pandemic has started, then wearing mask has become a norm. But unfortunately, there is a huge clinical studies are there. If you take all the conventional masks, be it N95, surgical mask, cloth mask, these are a perfect barriers, right? These are highly filtration barrier based they work. That's how they prevent all the bacteria and virus exposure. But there's a large clinical studies have done last year to show that actually bacteria and virus can accumulate on this fabric, on this mask, and can live up to almost 7 days. It can remain active up to 7 days. So that means we tend to touch the mask and while adjusting and so on. That means there is a possibility that we can touch and we can get exposed and contact based, or if you just discard them, it can spread out, right? So then that's where we thought can we develop a system which not only just filtration based, but which should able to kill the virus and kill the bacteria once it comes in contact. Okay. So, but first we wanted to see how much bacteria can really accumulate on these ones, right? So test that this is a typical agar plate assay where, this is without bacteria. And when you put the bacteria in a plate and you grow bacteria, this is how opaque you look, right? Now, what we have done is we have given volunteers this N95 mask, surgical mask, and cloth mask. We asked them to use for 6 hours. And after they use, and we take this mask and just touch this agar plate for a minute and remove. And after 24 hours, we see the growth of bacteria. As you can see here, okay, and all type of masks, they harness high amount of bacteria. In fact, cloth masks are even worse, they harbor even more. So, which is clearly suggesting that indeed, this bacteria can remain active and remain alive on the fabric. Although it can filter, but it can remain active there. So that when you touch, it can really get infected. So then what we thought is that to prevent that, we developed this small molecules. It's called germicidal molecules. It's a quaternary ammonium salts. And we also developed the process to covalently attach these molecules on the fabric. And what they do is these molecules, whenever either bacteria or virus comes in contact, they integrate into their membrane and break down their membrane. They rupture their membrane and kills them. That's how they deactivate all the bacteria and virus. And once you have this fabric, you can stitch into mask and a gloves and clothes, or any form of clothes you can make it, right? So once we developed, then we quickly wanted to see whether it works, right? For example, same assay if you do, what we have done is with this new germicidal fabric, we develop masks, okay? And these masks, we ask people to wear not only 6 hours, we also ask them to wear 18 hours long. And after that, again, we did the same assay. And if you see, we don't see any bacterial growth at all. So which is clearly suggesting that I think as soon as bacteria comes in contact, this fabric can completely deactivate them so that there will not be any bacterial accumulation. And similarly, we did entire viral assay also, including SARS-CoV-2. We tested and we have shown that it leads to within 5 minutes of time, 100% virus is deactivated. So again, like if you see, like, is it a good idea? Yes, I think there was huge unmet need. It was a timely invention we needed. So we developed that technology. But this is where, like, I want to give a different perspective about patents and publications, right? So in fact, we didn't file a patent application yet based on this. Okay. And the reason is we wanted to keep it as a trade secret. Because we want to do the rapid translation to the industry because we are running out of time, and we wanted to keep this as know-how knowledge transfer. So we kept that everything, and we transferred to the company as a know-how knowledge. And it's interesting, we didn't do any official publication, but like based on this technology, when it came out in media and like the 2 journals, top ACS journal, American Chemical Society journals, written articles on this work. In fact, American Chemical Society, what they have done is they followed all the technology worldwide developed in response to the COVID-19. And they selected top 4 in the world, and this technology is one of the top 4 has been recognized. So that shows the actually impact of this technology. And then what we have done is we quickly formed a team, like because we also wanted to people who comes with the experience with the textile industry. So we bring them into the team, and we formed a startup company called Color Threads, and we formed in India and also in US. And then we transformed the technology. We made the bulk process has been done, and bulk manufacturing is done. And in fact, in 2020 itself, in October, November, we launched all the products in the mass. And not only that, we also partnered with Aditya Birla Group, Vanusen Group, and through them also we launched them, and so on. So just to summarize at our work, like as I mentioned, like our overall effort in developing biomedical technologies. In the therapeutic biomaterials, we could able to develop disease-responsive biomaterials which could be used for protecting transplanted organs and the treatment of arthritis and diabetes. In the prophylactic biomaterial, the preventive biomaterials, I've shown like how we can use this as a preventing pesticide exposure, and as well as like preventing potential viral and bacterial infections. And today I didn't have a chance to talk about it, but like we also in the chemical biology program I couldn't talk, but we also developed this small molecular based classical drug discovery methods. We use medicinal chemistry and develop small molecules for the treatment of inflammatory bowel diseases like colitis and Crohn's. Maybe some other time we can discuss about it. But as I mentioned again, like we are not only just developing technologies, we're also equally interested in participating in entrepreneurship programs and translate them through the tech transfer. With that effort, as I mentioned, like just to summarize, through therapeutic biomaterials, we could able to put that platform technology and start a company in Boston in Allevia Therapeutics. And based on our prophylactic biomaterials, that's like only for anti-pesticide based technologies, we formed this CPO Health, which is located in Bangalore. And also based on our antiviral and germicidal technologies, we formed this Color Threads company. And in fact, for the chemical biology program, as I mentioned, the drug discovery company, based on that, we also formed another startup 2 years back in Boston, where we are doing entire further medicinal chemistry or the IND enabling studies we are doing to engaging with the FDA to go for the phase 1 clinical trials soon. So these are our efforts in the translational science. But again, this entire translational science entrepreneurship activity is a completely additional effort than our classical research effort, right? And the question is why we need to go all out of the way to learn and put that effort to do that, right? So then, but if you want to truly measure the impact of the translational science, and it not only just gives you the publications and patents, which are like normal reward from your academia, but if you do live really successful translational science practice, you can create jobs, right? Through our 4 companies, and also I have another company that based on my postdoc work I started when I was in Boston. So through all these 5 companies, we created more than hundreds of jobs now. And also, if you take a cumulative valuation of these companies, they are the worth of more than 2,500 worth. And this entire money, again, we can reinvest into developing more technologies and so on. So, but more importantly, what we see is the improving the quality of lives of people. The idea of what you thought and which came to the market, which people use and feel safe. I think that is the immense motivation of what a translational scientist can experience. In fact, I can't tell you enough, wherever you randomly walk in the Bangalore, you see people wearing the technology what we developed and feeling safe. I think that's the feeling what it gives us a lot more strength to go and develop more technologies and so on. So with it, I would like to stop here, and I would like to actually acknowledge wonderful group of PhD students and postdocs and master's students who did extensively working hard to develop these technologies, and the various platform technologies the group of people have worked. And I also would like to thank all our collaborators with both in India and elsewhere. And I would like to thank all our funding agencies for their generous funding. And I would like to thank all of you for your kind attention. I'll be happy to, take any answers, any questions you may have, happy to discuss further. Thank you so much for your time.
Thank you very much, sir, for sharing your time, valuable contribution in the scientific research. And it's truly, sir, we experience today, and this is a unique opportunity for all of us to understand translational research and how the innovative ideas can be generated, and we can transfer, we can reach them up to the clinic. And congratulations, sir, for all your outcomes from this research, big startups, international startups, and generating the jobs for the youngsters. So we shall we start with the question answer session, sir?
Yeah, please. Thank you.
So we have one question in chat box from Sentili Raja. Sir, how to prepare hydrogels? What materials may use to prepare it? How to fuse drugs into gels? Is hydrogel formulated drugs are beneficial alternative to already suffered arthritis patients?
Well, I think it's a pretty general process. It is there are 2 types of hydrogels are known, right? One is you can use the polymer-based hydrogels. Typically, polymer-based hydrogels you can make through either cross-linking, either covalent cross-linking or ionic cross-linking, or some of them they form a simply swelling process. Okay, but we primarily work on self-assembled hydrogels, meaning you take a single molecule, and when you put it in the solvent, what they do is they use these intermolecular interactions hydrogen bonding and van der Waal interactions, pi-pi interactions. By systematically we incorporate into the molecule, so that all these molecules come together, the millions of molecules comes together, they form these hierarchical structures, right? That's how they form these nanostructures and micron structures and bulk gels. Okay, by tuning this amount of intermolecular interactions you can have, one can modulate your self-assembly process to make your hydrogels and organogels and so on. And during this self-assembly process itself, if you can incorporate the drug, when they're forming self-assembly itself, they capture the drug and encapsulate them in, and they form the gels. Because once you form the gels, you can't put the drug because it's like a diffusion. It's a very inefficient process. It doesn't go. So that's why we tend to encapsulate while forming the gel itself. That's why we don't see much non-specific release. Otherwise, it can easily diffuse without having enzymes, right? So that's why the self-assembly is very critical here, how you modulate that.
So in the same line, I would like to ask one question.
Please.
So how we can characterize those materials? For example, we self-assemble the small molecules in these gels, or if we conjugate with the labile bonds with some gel material, how we can chemically or how we can characterize those materials?
Yeah, there are various analytical tools are there. You can use for example, rheometers. Their mechanical properties can be studied by rheology. And you can also use electron microscopes, such as scanning electron microscope, and also transmission TEM and SEM can be used. So these are the regular analytical tools one can use for that.
So another question is, so is there any theory on immunosuppressants used locally after bone marrow transplant, and to prevent its side effect or transplant rejection?
Bone marrow transplant, I'm not aware of that. At least I didn't come across any examples of having localized therapy for bone marrow transplantation. One of the biggest challenges, for the bone marrow transplant and all, you can do only one-time injection there. Only during the transplantation, during the surgery only you can do. Repeated times, it becomes super invasive, right? Super painful to give repeated times bone injections. So that's why localized therapy might not be very valid for the bone marrow transplantation. There it might be systemic transplantation, systemic immunosuppression might be better.
So few questions from our end that when we're finding the unmet need for any research, so how should be our approach is that the product or the process that we are developing, it should be for the large population, or it should be for a small population, but where there is no treatment existed, like for the rare diseases?
No, it is equally important. Okay, so as I mentioned, I don't think we merely decide based on the number. Okay, because what you have to see is is there any technology already available there? Okay, it's either doesn't matter whether it's a large population and small population, but whether is there any technology already is there, or there are plenty of things there, but we can also make another one. Okay, so then how much impact that is going to make? I think that is what the critical decision one need to make. If there are there are 10 other ways to treat the disease already, and of course this could be another 11th method, but how much impact that is going to have, that we need to see. Because you see, any drug development, right, it's a huge process, right? You're talking about 10 to 15 years timeline, because any drug development, if you take right from the discovery to going through entire clinical development, clinical trials, and coming to the market, you're talking about 10 to 15 years time, and hundreds and hundreds of $1 million investment, right? So one need to see if you really want to bring it to that stage. If it is only another one more drug with a little more improvement, you wouldn't be able to raise such money. You don't get the investment to take to the till end. So although as a conceptually one can work and develop, but the translation becomes super difficult. So that's where we need to see like whether if you give this solution, whether you can raise hundreds of $millions to develop that further in the clinical. That's the decision one need to make. So in fact, rare diseases becomes super popular, although the number of people are less, but the technology is generally very rare, like very less treatments are available for the rare diseases. So that's why like most of the companies also, they focus a lot on the rare diseases. Not only that, like there's additional benefits, because if you go to the regulatory approvals and all, you have much faster regulatory approvals, and that your clinical trials also a little shorter, less number. So your development process becomes smaller for the rare diseases. So your overall translation time might be smaller to go to the market. So we need to see all these factors and decide.
Thank you very much, sir. Sir.
Yeah, sir, actually, there is one thing in the similar lines which you just said. So actually, we are working on stem cell research also for autoimmune diabetes and the complications related to it. And we actually got very good results in the preclinical phase for diabetic foot by using the mesenchymal cells. And preconditioned cells also we are using it, and for even for the reversal of hyperglycemia and type 1 diabetes also we have got good results. But all these are in the preclinical studies right now. So whatever results we have are in the preclinical research. So like as you said, so the next step to get it to get it translated to the clinic or to the patients for the clinical application, you suggest that should we approach the companies who are working here, or how we should take the next step?
Okay, so the first thing you need to make a decision is like whether is this a technology should be simply licensed. I think you must have filed the patents and all, right?
Yeah. In the process it is, yeah.
Okay, so one thing you need to see is like, is it for this technology is a simple tool, like, go to the existing company, just translate this technology, license them so that they can do the further development, or whether this technology can be used as a platform so that you can form a company and develop from there. Okay.
Okay.
That is what we need to see. You have to analyze that. But if you go to the company to license it, right? One thing you need to do, you might have to do a lot more for the development, because just conceptual idea can be fine. Proof of concept is fine, but whether it is industry ready, we need to check that, because if industry want to take the license, they want to have much more advanced level. Okay. And there are something called de-risking process. Okay, but for each technology is a different criteria you will have for the de-risking. So for this particular one, so you need to see like, what are the checkpoints you need to cross so that, they are comfortable to take it. Because too early, if you go like, they will not be like interested, saying that, okay, it's too early, we will not. Then you need, but like even further, you have to de-risking, then you need to find the funds to see like what are those steps you need to take care of so that industry will be ready to license it. So that's the decision I think one individual need to make, because if you go through other route, you forming company and all, then you need to see like whether you're okay to go and form the company and find the team and raise the money. This entire other group of activities one need to start looking for, whether you'll be able to dedicate that time and do that. So if you're ready to do that, I think, why not? One can experience. So that's each technology one need to make that decision individually, case-specific manner.
Thank you so much, sir. It was really wonderful to interact with you, and even the presentations which you have shown, the kind of explanation and this idea of translational research is really great.
Thank you.
Thank you, sir.
Sir, one question is from Dr. L.P. Kuttapalli, ma'am. For psoriasis, if we target metalloproteases, can you use some insights?
I am not very aware of what is the role of metalloproteases in the psoriasis. I don't have much idea about that. Because inflammation produces these enzymes, but I'm not sure what is the role of these enzymes in the disease progression. I don't know. Actually, I need to read about it.
So, like in your one of the research projects where you design and develop the responsive drug delivery systems, so where the inflammation triggered drug release is possible. So on the same line, can we design some delivery systems where the drug will release in response to the level of hormones or with the level of neurochemical transmitters, because their levels are also responsible for the disease conditions.
See, conceptually, it is possible. Okay, so that's what we have shown. But like this is where it becomes case specific. Okay, then you need to see exactly what biomarker you want to have as a trigger stimuli, and then by looking at the structure and the function of that, okay, then you need to then design a material which can be responsive to that particular biomarker. Okay, say if it is a neurotransmitter, if you're saying that, say acetylcholine, some small molecule, so it might become difficult because if the acetylcholine, if the small molecule, how that can degrade any biomaterial to release that drug. Okay, so that's why like there are two critical things one need to follow. One, identifying right biomarker, and then custom designing the material which can be responsive to that biomaterial. Okay, these two steps need to be done each disease individually. There's no one general solution for that, but conceptually it is possible. Yes. The best is diabetes, right? Like if you can develop material for where the level of glucose can modulate the release of insulin, that's a fabulous one. So people have been trying that for decades, in fact, right? Like there are a couple of like in vitro and animal experiments were done, but like nothing really translated yet. So that's why it is possible, but like it's a case specific manner one need to optimize for each disease separately.